mentally at least, if not physically.
All right, well, welcome back. Got all the quizzes up now, and thank you guys for those of you that have taken them. I'm not really going to focus on any of the answers from the quizzes since not everyone has finished those. But I think that probably the next 2 class periods we'll be working on review. Again, this is my first time teaching this class, so I'm sorry for probably making too much material on this second unit test. It'll be a big test. Probably should have split that into 2 different tests. That's my fault. So just bear with me. I've got a pretty large review planned for us, just highlighting really the pertinent information from the unit. And I think it's going to take us a couple of days to get through it. So I'm really going to highlight the big stuff here. All the quizzes are up and ready, so definitely go ahead and take those. Also, I know that you guys have other tests too and a lot of things to be working on, thinking about, but try to begin some work, especially on the research for your crop project. And again, if you have not signed up for your crop project, please do that as soon as possible so that I can get an example up for you. I don't want to do an example of a crop if someone else is going to choose it. So if we don't see those up in the next couple of days, then I'm just going to have to just go ahead and just do my example anyway. Well, let's get into the review. Let's share my screen here. All right. You guys see the PowerPoint there?
Yep.
Okay. Just making sure. Technology is not necessarily my strong suit. So, all right. So let's start off with Chapter 6 here, looking at the structure of higher plants. And just a few highlights here. Major food, fiber, wood, and ornamental plants belong to these 2 main classes, gymnosperms and angiosperms, but not every potential crop does. We could look in ornamental plants. We've got like ferns, for instance, that are ornamentals, mosses that we can use as ornamental plants. These are not the be-all and end-all of crops, and really we can get really creative with our cropping systems and produce different crops. And personally, I think that's a big thing that we need to focus on as agricultural producers. What is your niche? Don't be boxed in by the 2 main classes, even though the vast majority of crops are within those. We're looking at the flowering plants. We have 2 subclasses, and of course, most of our crops fall into the flowering plants, not all of them again. But it's important to note the difference between monocots and dicots. In the monocots, have 1 cotyledon when the seed germinates and pops up, that first seed leaf. There's just 1 for monocots. There's 2 for dicots. Other big thing is that monocots in their leaves have parallel veination. The veins of the leaves are right next to each other and come from the bottom of the leaf up, whereas dicots have net veination. Moving on to the protoplast. Big thing here I want to talk about is the cytoplasm, which is basically where all the organelles are kind of floating around in the cell, kind of holding everything together, bounded by the semi-permeable plasma membrane. And the flow through the cytoplasm is known as that cytoplasmic streaming. Another important thing to recognize about the cytoplasm is that there is some DNA stored in the cytoplasm as well. For a long time, it was thought just to be in the nucleus, but there's some DNA also found in the cytoplasm, and that is inheritable information. Speaking of the nucleus, that's what we kind of consider the brain of the cell, and inside of the nucleus are the chromosomes, which contain these long lengths of deoxyribonucleic acid, DNA, the good stuff. And as I mentioned, it's also found in the mitochondria, in the cytoplasm, as well as, to a lesser extent, but a little bit in the chloroplast as well. The units of heredity in plants, in the DNA and in the chromosomes, where the genetic code is stored. Let's look at the plant tissues. We got the meristem, of course, and as you know, the meristem or the meristematic tissues are where these cells are actively dividing and, maybe more importantly, developing and differentiating into different types of tissues and then into different organs. So the magic happens in the meristem. And we have those apical meristems, not just in the shoot, the above ground terminus of the plant, but we also have those in the roots, which could be in a tap root or in a fibrous root system. They are also at the termini, right? The ends of the roots are where we have that meristematic, that differentiation taking place. In the roots, it's saying, well, are you going to be more of a root hair, or are you going to be a root cap, etc. Then there are also the lateral meristems, especially in woody plants, that provide the secondary growth and kind of the girth of stems. And these meristems are the vascular cambium, which is responsible for producing new xylem and phloem, and the cork cambium, which produces bark in woody plants. Y'all slow me down if I go too fast or anything, or if you have any questions. It's just, this review is meant for you, so speak up if you need any more information about any of this. Moving on, we got another thing called the trichomes, pictured here. The trichomes are often where we have kind of secondary chemicals stored. These are also very important for secondary products coming from plants. Some of these permanent tissues are extremely important. Of course, the xylem, which is responsible for moving mostly water as well as nutrients dissolved in the water throughout the plant, and generally from the roots to other parts of the plants, but can also move water throughout the plant as well, up and down movement. Phloem, same deal, could be up or down, but generally mostly is conducting food and moving the carbohydrates and other metabolites from the leaves, where they're photosynthesized, to stems, flowers, roots, and other organs. The xylem and phloem, extremely important for moving water and nutrients throughout the plant. As I mentioned before, with the monocots, we have often more strap-shaped leaves, more narrow leaves with parallel veination. Talking about water and xylem, we have this whole movement of water through the conduits of a plant from the roots and throughout the leaves as the process of transpiration, which also is very important, helping to regulate leaf temperature, as well as giving turgidity to the plants. Something I wanted to mention here, on another kind of category of plants, we have dioecious or monoecious plants. And if we break down the Latin of those words, dio meaning two andecious being technically house, so two houses, that would be plants that have separate sexes. Not a whole lot of our crop plants do have this kind of dioecious characteristic, but there are some that do. Spinach, for instance. some other things in that same family, like beets, chard, or dioecious. And it's important to recognize that if you are, growing dioecious plants, in order for it to produce fruit, you have to have both male and female plants. There's an interesting way to get around that I'm actually researching in pawpaw production, actually grafting male parts onto the female, so artificially making a plant. The other category, which is monoecious, one house where both sexes occur on the same plant. So know the difference between monoecious and dioecious. When we're looking at plant growth, we look at the size increase of the plant through cell division and enlargement, as well as the synthesis of new cellular material being created into organs and tissues. And these stages of development begin with a seed germinating, and then the growth of the vegetative organs and tissues, as well as the initiation and maturation of the reproductive organs and tissues, fertilization, pollination/fertilization, seed development, and then maturation of the plant, and finally senescence and death of the plant. And we see this in terms of division, elongation, and expansion causing shoot growth in plants from the meristematic regions of the plants, again, both in the shoot and in the roots, as well as in the lateral meristems and the thickening trunks too of woody plants. When plants mature, though, they switch. The vegetative apical meristems then become reproductive meristems and can produce floral parts. And as we'll get into here in a little bit on the review, talking about how those floral parts are often in that switchover as a result of hormonal changes. Kind of like how it is with people too. So what are these signals, these hormonal changes that regulate this action? They're these plant hormones, including auxins, gibberellins, cytokinins, and ethylene, and some others, but those are the big ones we need to note, which work by actually activating and deactivating different gene transcription. So on the genetic side, genes can actually be turned on and off by these endogenous hormones inside of plants. Let's go to the light for a second. Photosynthetically active radiation, or PAR, which is also visible light to people, is the wavelength that is the most important for photosynthesis. And that is from 400 to 700 nanometers in the wavelength if we're looking at that electromagnetic spectrum. The Roy G. Biv. Another important thing with light is the angle of incidence, where basically how that sunlight is striking the plant. And of course, that angle of incidence at the the longest day of the year, the summer solstice, we get that 90-degree angle of incidence. And the rest of the year, it moves in either direction. And pretty soon here, we actually will be running into the point where it's usually around November 15th for our area, where light basically becomes a limiting factor for the growth of plants. And even if you're able to maintain temperatures, if you don't have light, they're not going to grow. So in order to get things to grow that time of year, you have to provide some artificial or supplemental lighting. So again, there are optimal temperatures for maximizing vegetative growth and really even getting plants to grow at all, as well as flowering. And that growth often occurs between 39 degrees and 50 degrees for temperate region plants. These are generally the limits of plant growth, but it's not the be-all, end-all limit for all plants. there are many plants that can grow well above 50 degrees and some that can grow below 39 degrees. So just keep that in mind. There is a limit, though, on high temperatures. Of course, high temperatures can be bad as well. There's a maximum temperature for plants, and above that temperature, we have destruction of cell protoplasm. And we can get solarized or sunburn leaves, especially heat damage when we have relative humidity dropping and we have drying winds. Of course, we also have, on the opposite end of the spectrum, lower temperatures causing what we've been having a lot of lately, frost, that actually causes the rupturing of cell membranes and walls, and then subsequently the drying out of cells, which we can prevent by basically putting a barrier between the plants and trying to prevent the radiant heat transfer from the leaves to the sky. Cold temperatures are not necessarily a bad thing. In fact, they can do good things for things such as seed germination. Stratification is that cool, moist period that seeds need to actually overcome dormancy and allow germination. Also, for flowering, we have cold induction of flowering, known as vernalization. And in some crops, we have a chill, especially perennial crops, we have a chilling requirement, a number of hours that these plants have to have below a certain temperature in order to break dormancy. Moving on to water, cells are turgid with water. Water is what causes the turgidity of cells and allows plants to stand up through transpiration, basically. And we can actually withhold water to keep plants smaller in some cases. Why would you want to do that? Well, mostly for ornamental purposes. Two important gases for the growth of green plants, CO2 and oxygen. Mostly the oxygen is needed mostly in the roots, but above ground parts needed as well. Carbon dioxide, as we know, is extremely important for plants for photosynthesis. Also for stomatal opening, and oxygen being important in the respiration of plants and driving these biochemical reactions, which we'll talk about more in here in a little bit. Seed germination occurs 3 major stages: water uptake / imbibition, and then an increase in the biological activity, which we're really seeing as the utilization of food from the endosperm and that transfer of that food to the developing plant, and then the radical or the first root and then shoot emergence. What do roots do? Well, they anchor the plants, they absorb water and mineral nutrients, they conduct that water throughout the plants, and in doing so also provide turgidity. They also store food in some materials, especially in kind of more modified stems, but they're underground, so we look at that as part of the root system. The root system and shoot system have a balance, and as the top of the plant grows larger, the leaf area and water loss through the transpiration tends to increase, and that increase in water loss is made up by water absorption from an increasing root system. So there has to be some sort of balance there in root and shoot growth. Shoot growth, of course, as you all know, it can be determinate or indeterminate. Determinate being more kind of bushy growth, and it often matures crops all around the same time. Indeterminate tends to grow throughout the growing season and is often more vine-like growth. So indeterminate tomatoes there on the left, and then a determinate bush type of poinsettia there on the right. 3 life cycles of plants. We've got annuals, 1 growing season. Biennials, 2 growing seasons. Not necessarily 2 years, but 2 growing seasons. One where they're producing vegetatively, and another one where they're producing florally, reproductively. And it's exposure to the chilling temperatures that trigger the hormonal changes for all plants, but especially for biennials. Then we have perennials, of course, which can remain alive for long periods of time. I say indefinitely, but really, I mean, all plants have a life cycle. They're going to die at some point, a lifespan, I should say. And in the perennials, they have kind of adventitious buds at the crown of the plant that are important for the expansion of the plant. Another thing to recognize is that we do grow herbaceous perennials here as annuals in many cases, like tomatoes, for instance. They're in their native region, they are perennials, but here we grow them as annuals. Lots of our crops are perennials in their land of origin. As plants develop, they can change. They can look quite a bit different in their leaf structure, stem structure, whether they are thorny or not. All kinds of things can happen as plants begin to develop. It's extremely important to note how those changes happen in plants and how they look different from the seedling to the mature stage of plants, to be able to ID them and know whether you got a crop or a weed in some cases. Most of our agricultural plants, most of our crops actually initiate or form flowers as a result of morphological maturity. So once they get to a certain height or a certain stage, they switch over to reproductive stages. And that is often determined by how much heat is received by the plant. And we can look at that in terms of growing degree days, again, which is actually recorded by many of the weather services. We got vernalization as any cold temperature treatment that induces or promotes flowering. We already kind of mentioned that, but do that again there, especially in the case of biennials. All right, fruit, grain, and seed formation all begins with pollination. And we have several different types of pollination. We've got self-pollination or cross-pollination. And that's important to note for how we might save seeds, how we might even grow our crops in many cases. Self-pollinating plants are self-fertile. They generally are perfect plants that have both the male and female flowers on the same flower. Self-sterile plants or cross-pollinating plants cannot set fruit from its own pollen, but often requires pollen from other plants due to incompatibility mechanisms. Often they are actually there to overcome things like inbreeding depression. Pollen transfer in cross-pollinating plants is chiefly done by insects and wind, although there are other organisms that can do that as well, and other non-living things too, like water. You have organisms like snails, slugs, birds, bats, other mammals, all kinds of things that can act as pollinators. When pollen lands on the stigma of the plant, that receptive part, it triggers the creation of a pollen tube. It goes down and actually fertilizes the ovules. That's fertilization. And fertilization can actually occur in some cases without the stimulation of pollination and fertilization. That is known as parthenocarpy. And that can actually be induced in some fruits by the application of some hormones like gibberellins and auxins. And of course, stenospermocarpy, we see that as basically fertilization without the formation of seeds. We get fruit but no seeds, and that's how we get things like seedless grapes. That's a good thing. In particular cases anyway. Accessory tissues in the flower are often involved in fruit formation, but the true fruit itself is just the enlarged ovary. And in most plants, only a small percentage of flowers tend to actually develop into fruits. And in some cases, we have too many of them developing into fruits that can cause damage to the fruiting crops. Or cause some fruits to be too small. Would you rather have a ton of little teeny tiny fruits, or would you have fewer larger fruits? Well, it tends to be that consumers produce or tend to prefer fewer but larger fruits. So to overcome heavy fruit set, a lot of times we will actually remove excessive fruits at early stages through a process known as thinning. And in some cases, we actually have some self-thinning fruits, especially in a lot of our fruit tree crops, where they just drop on their own. We're going to look at some of the different hormones and how they affect plants. One of the big ones, though, that we see very often is the application of gibberellin to grape clusters to increase the fruit size. Another thing on fruits, remember the climacteric fruits. These are those fruits that can ripen after being harvested, and they can over-ripen after being harvested as well, right? So apples, apricots, bananas, tomatoes are just a few examples. And one of the ways that we can actually hasten that ripening can be to treat them with the plant hormone ethylene. I mentioned senescence. Of course, senescence is the beginning of the death of the plant. Plants senesce and then die. But senescence is not necessarily always a bad thing. It does some good things for plants. It's needed to switch energy allocation in plants. One way to kind of prevent senescence is through vegetative propagation. We can create clones, then technically we're like extending the life of the plant. And I believe I mentioned when we were talking about this, how there was a bonsai guy in the area who used to preserve the collections of bonsai for the U.S. and D.C., and he mentioned that some of these are like 3,000 years old. Well, they're not really actually, been preserved for 3,000 years. They've just been taken from plants that are very old. So that's kind of what we see. And in some of these cases, we have clonal cultivated varieties that really have the same genetics and have been just cloned for long periods of time. Thousands of years in some cases. So senescence, of course, is caused by the natural maturity of the plant, the aging of the plant, but can also be caused by attack of pathogens and other environmental stresses. And as I mentioned before, it's not entirely just degradation. In some cases, we have particular proteins and messenger RNAs that are synthesized only in senescing tissues. So it's important in completing the life cycle of the plant. And it is hastened by the transfer of these stored nutrients to the reproductive parts. So if we actually remove those reproductive parts, such as just harvesting fruit, then we can delay senescence. And we do that very often in crops like summer squash or cucumbers, beans even. By harvesting them, we're delaying senescence. If the plant completes its goal of life and actually produces mature seed, then it will begin to senesce on its own naturally. Let's talk a little bit about some plant hormones. Plant hormone being a natural substance that acts to control the plant activities. Plant growth regulators include these plant hormones and can be natural or synthetic, and can be applied exogenously as well as endogenously to the plant itself. We got 5 traditionally recognized groups. Know them: the auxins, the gibberellins, the cytokinins, ethylene, and abscisic acid. And just a little bit about each of those. We have auxins occurring in meristems and enlarging tissues, and they are very important in creating apical dominance. The growing tip is where most of the auxins are stored in the plant. Gibberellins are important for stimulating cell division, cell elongation, and can also control some enzyme secretions as well. It can affect fruit set and fruit growth, as well as the maturation, the ripening of fruits as well. They can also be used to overcome dormancy in some seeds and buds. And of course, I had to go back to the grape example, one of these classic examples. The application of gibberellins at a particular level here vastly increases the size of the grapes in a cluster and makes for a much better crop. Cytokinins interact with the auxins to actually influence the differentiation of tissues. So it's the balance of cytokinins and auxins that affect shooting and rooting. And there are over 100 known natural and synthetic cytokinins. Lots of them out there. Ethylene is mostly used as a gas, which moves throughout the plant, and you don't have to have a whole lot of it to actually have the effect of ethylene, which is used for fruit ripening, flower initiation, sometimes changing the sex expression of the plants, degraining of climacteric fruits, aiding harvest, and growth regulation as well. But it can also harm plants, especially when we don't want them to continue to mature. So causes rotting, right, in many cases. And there are many things that can increase ethylene, natural and unnatural. Abscisic acid is very important for the regulation of the processes in seed development and senescence of the plant. So it can also be important as a stress hormone. So abscisic acid, there you go. It causes the dropping of leaves, for instance. Let's move on and talk about some metabolites. Metabolites are these that are formed, through photosynthesis and then production of these secondary chemicals, and they are the basis for nearly all plant nutritional needs. Very important in many different plant processes. The creation, the synthesis, and metabolism of carbohydrates and sugars all begins with photosynthesis. Plants, of course, depend on carbohydrates for structure and energy storage. And what is a carbohydrate? Well, basically it's carbon with water, right? It's hydrated carbohydrate. Pretty cool. 1 carbon, 2 hydrogen atoms with an oxygen atom is the general formula of a carbohydrate. Of course, we have the most common carbohydrate, glucose, being C6H12O6, which is not just the most common, but also the most important of our carbohydrates. And only one change in the structure of a starch, a particular type of carbohydrate, is needed to make the structure of cellulose. Even though they have massive differences in plants and how they're used, it's just the position of one hydroxide molecule in the structure of that carbohydrate. Kind of crazy. So it's some of these differences here. Starches are slightly water soluble, can be broken down by various organisms to the simple glucose molecules, so they're very important for nutrition. Cellulose is insoluble in water and provides the rigidity for cell walls. It is not really digestible, mostly indigestible by animals. So not as important in nutrition, but very important for the structure of plants. Look at some lipids. Fatty acids and lipids are very important in cell membrane structure and also for energy storage, especially in seeds. Also important for human nutrition too. Fatty acids can be saturated or unsaturated, as we know. And there are many different materials, such as fats, waxes, and oils. They each have different physical properties that all belong to this class of compounds known as lipids. These types of fatty acids that are found in the membranes of plants can affect the ability of the plants to withstand cool temperatures. Fats act kind of as an insulator, not just in plants, but in me too. Why tropical and subtropical plants suffer chilling injuries at temperatures that are well above freezing is due to the fact that saturated fatty acids will start to solidify in cool temperatures. So even though it's, not like dying, it's not frost damage, you can have chilling injury just as a result of the types of fatty acids that are in the plant. Again, they're important, especially in oils, for human nutrition. So you get some proteins, which are just long chains of linked amino acids that create these very complex three-dimensional molecules. And they all have a very common structure that consists of nitrogen, hydrogen, carbon, and oxygen. But they differ by the wide variety of chemical groups that are attached to their common structure. Proteins, again, are composed of amino acids, which there are 20 different amino acids found in proteins, 8 of which are essential to life on Earth. Methionine, valine, leucine, isoleucine, threonine, phenylalanine, tryptophan, and lysine are 8 essential amino acids. They cannot be synthesized by animals. They have to be obtained from, at least initially, from a plant source. There are various storage protein structures in seeds, and that's what makes seeds so important for human nutrition, as well as animal feeds as well. They're generally 5 up to 25% of the dry weight of seeds. It's the balance of essential amino acids in these proteins, which we cannot synthesize, that is critical for their nutritional value. And in plants, the nitrogen is needed for the proteins. Again, one of the nutrients that is taken up by roots in the largest quantities and one of the most often deficient nutrients that has to be supplied by fertilizer. ATP, adenosine triphosphate, is one of the most important molecules in all plant metabolism, or really all metabolism, because of its importance in plant metabolism. And that's due to the energy stored in these phosphorus bonds. That release energy provides the energy for other biochemical reactions in plants. We also have organic bases, which their importance is in the function in creating nucleic acids. We look at RNA, ribonucleic acid, as this very important chain-like molecule that is kind of this transcripting factor for DNA. It's used as a template for the production of DNA. In DNA, we've got these organic bases that are A, G, T, and C, adenine, guanine, thymidine, and cytosine, which are a little bit different in RNA. These make up these long chains of DNA molecules through the base pairs that make up chromosomes and carry the genetic information in order to create proteins, which drives life that builds and maintains the organisms. Just real quick, looking at the secondary compounds that are not part of the main processes of the metabolism in plants, but are often used by humans for several different means, often for medicines or in some cases for recreational drugs, such as the cannabinoids, things like morphine, cocaine, nicotine, and caffeine. These are all secondary products that might have some use in plants in protecting the plant itself, but really are more important for medicinal or recreational uses for humans. We also have alkaloids, different secondary products that are often used by plants, again, as protectants or deterrents, often to things that are like herbivores, things that are eating on the plants. We've also got terpenoids, which if you guys still have some leaf color over there, that's some of the things that we see actually causing pigments in leaves. These pigments actually serve as a protective role in the chloroplasts. Some of them actually also are used for protective roles against insect invasion in plants too. The plants that we cultivate all originated from wild plants, as we talked about very early in the class, but most of them appear very differently from their wild relatives. And these differences have been created through recombinations of genes and then the selection and redistribution of these heritable traits through the generations, through breeding, basically. I don't know why that did that twice. So important to note, and we'll see different picture of this later on, but again, Vavilov's centers of origin and domestication of food crops. Where do these crops come from? These centers of origin are also the centers of diversity and where we need to look at preserving the germplasm, obtaining that germplasm from our crops. A little bit about chromosomes. The number of chromosomes is the same in all the vegetative cells of an entire plant species. And we look at that as the 2N or diploid. Again, all of our vegetative cells in plants or all of our regular mitosis cells in humans are diploid. They've got complete sets of chromosomes. But in the sex cells, they are reduced in half, known as haploid or 1N. The number, size, and appearance of chromosomes can vary considerably. To date, we have chromosome numbers known for most plant species. Again, chromosomes are composed of DNA and RNA and proteins, basically. And it's this active genetic material that in DNA that replicates itself and transmits information to create proteins and drive life. Again, made up of many repeating nucleotides. In the base pairs, the adenine, thymine, guanine, cytosine, held together by hydrogen bonds in this double helix shape. During cell division, the chromosome split. The DNA molecule replicates itself by unraveling and separating at these hydrogen bonds, and then becomes a new double strand when it comes back together. DNA is basically self-perpetuating genetic code, but it's the RNA that actually controls the cell growth. DNA being double-stranded, RNA being single-stranded. In RNA, we have uracil replacing thymine as a base, kind of as that transcription factor. As, again, RNA being the template for DNA. Moving on, structurally, a gene is a sequence of triplet organic bases along a DNA molecule. So that's the ultimate hereditary unit for plants, animals, all living things. And genes on the same chromosome are said to be linked together, and they can move from one generation to the next as a unit. And that linkage is not always perfect, and sometimes they can break apart and exchange. But the linked genes often work together to actually control different traits. So it's often not just one gene. In some cases it is, but in many cases it's several genes that control, several linked genes that control different appearances in plants. Genes are called alleles, or are termed alleles to each other if they occupy the same position on a homologous chromosomes and affect the same trait. So again, alleles can be the same or they can be different. The same is called homologous. Different is homozygous versus heterozygous. Or they can be dominant or recessive. Dominant would cause it to be expressed, whereas a recessive gene only is expressed if both alleles are recessive. Cell division just regularly occurring is called mitosis. And again, that is kind of our 2N, our diploid, typical cell reproduction that is going on. Non-sex cells. When we have sex cells reproducing, we have reduction division, the haploid division, meiosis, which allows heredity to take place. As plants grow, there can be errors, of course, because there's so many different cells dividing all the time. So there are transcription errors that can cause mutations in plants. And the majority of these mutations are negative. They do not positively affect plants, although some of them can be good and some of them can, actually create kind of evolutionary differences in plants. Different alterations that can make cool new features of plants. Polyploidy is something that we have to examine because polyploidy is the condition in which individual plants have more than 2 sets of homologous chromosomes in their vegetative cells. And the reason that polyploidy is so important is because it cannot, if you have a diploid organism, it cannot breed with a triploid organism. They might be able to produce fruit, but they won't be able to actually produce seed. And we talked about the whole example of the seedless watermelon and how it uses that whole idea of polyploidy in order to be able to create a fruit that does not have seeds or does not have well-developed seeds anyway. Recognize the difference between genotype and phenotype. I think you all know what that is already, though. We're looking at the genetic makeup for genotype and then the actual physical appearance as expressed in interaction with the environment as the phenotype. When we're breeding plants, the big thing in breeding is selecting plants. Selection is huge. Looking at the desirable characteristics and intentionally or unintentionally crossing them to produce offspring that has all of the desirable traits that we want. That's kind of the basis of plant breeding. And often what we do with plant breeding is actually initiate some cross-pollination, whether that just is planting 2 different types next to each other or actually doing hand pollination as they're doing here in this picture with carnation. Whenever we cross 2 different varieties or species of plants, then we create a hybrid. And hybrids tend to be more vigorous than either of the parents. Hybrid seed produces an F1 generation that has 2 different yet homozygous parents. But then the F2 generation is going to be heterozygous, and there's going to be a lot of variation due to the segregation of these different genotypes and phenotypes in the plants. So open pollinated crops, of course, can be saved very easily, and they're reliably pure. Hybrid crops, on the other hand, cannot be saved to have reliably pure plants. They're going to be different from the parents. And we're out of time, so I'm going to end there on this breeding slide, and we'll come back next week and finish our review. So anybody have any questions, comments, or concerns? Are you overwhelmed, underwhelmed? Is everybody good? All right, well, if you have any questions or concerns, feel free to contact me. I'm going to go ahead and put this half of the review up. Yes, you said.
Quizzes for chapters 12 and 13, the water and I think it was protein or minerals. Are those up?
They are up and ready. All quizzes are open and ready for you to take.
Okay.
All right, you guys have a great day. Good luck on your test.
Okay, bye everybody.